second laureate for optogenetics is Dr. Karl Deisseroth, who was really focused on the challenge of determining how electrical activity in different neuronal subsets can control behavioral responses. And he adapted the light-activated proteins that you've just heard about to allow individual neurons to be controlled with light in real time during animal behavior. And Dr. Deisseroth's group designed the necessary tools for targeting opsin and light to control circuit elements and apply the final resulting method, called optogenetics, to discover principles of brain function. And he has been and continues to be a leader and driver of advances in optogenetics as applied to neuroscience. Dr. Deisseroth is the D.H. Chen Professor of Bioengineering and Psychiatry at Stanford University and an investigator of the Howard Hughes Medical Institute. And I'd like to invite Dr. Deisseroth to the stage to tell us more about his work.
All right, great. Well, thank you, Janet, and thank you all for being here. It's a tremendous honor and a particular pleasure and a privilege to share this honor with my friends and colleagues, Peter and Ed. And this is something that has, as Peter captured very well in his talk, is really a confluence of basic science dating back even more than a century ago to very recent advances in understanding structure and function of algal proteins. And this has moved very quickly. Even as recently as 2011, this was our level of understanding of what the channel rhodopsin looked like. We didn't know exactly where the pore was. There was some prominent discussion that the pore might be formed at the interface of two 7-transmembrane proteins, and with that level of mystery as to where this near magical light-gated pore actually was, we couldn't do engineering of the pore, we couldn't transform it for new kinds of function, and it was also just a fundamental mystery that was exciting to solve. Peter showed this image. This is the product of a great deal of work over many years that has now given us an understanding of these amazing proteins. The Gairdner Foundation is recognizing both the basic science of understanding how these proteins work and their applications to neuroscience. And I'll talk about each of these aspects in the first and second halves of my talk. As Peter mentioned, there were many people who wanted to do something like this. In addition to Roger Chen, Francis Crick also wanted to do this, and he didn't get as far as Roger in realizing what a good strategy might be. In 1999, he did say that for neuroscience, we need to be able to control neurons specifically, and that light might be a good signal with its natural properties of being able to enter biological tissue non-invasively, its speed, but how to convert that into something functional was the question. But as Peter also mentioned, many people, many different groups eventually did try the microbial opsins in various ways, including the channel rhodopsins. And the essence of this, what became optogenetics, is that one gains incredible leverage that electrical and magnetic interventions in terms of understanding how nervous systems operate, these cannot distinguish individual cell types, which may do completely different things but be right next to each other, because every neuron responds to electrical signals, having these electrically gated channels in their membrane. The essence of optogenetics, and particularly, and this is a crucial point, with these single genes that encode single proteins that encompass both the light sensation and the ion flux in a single gene, this is crucial for the basic principle of optogenetics, to be able to efficiently and robustly in a versatile fashion target, using genetic or anatomical tricks, to cells or cell types of interest, the necessary transducer of light into current. Pioneering work from other folks had tried other strategies. Gero Miesenbach, Richard Kramer, and other folks had tried and succeeded in conferring photosensitivity onto cells in a light-gated and genetically targeted way, but these approaches required multiple components, and so didn't fulfill the full promise that these single-component proteins allowed. Now, of course, the opsins encode genes that don't look like this. They have a great deal of interesting structure, and it was very productive to understand that structure in great detail, both with obtaining crystal structures and with molecular dynamics modeling and other approaches. As Peter indicated, we were able to speed up the cycle of the photocycle of channel rhodopsin, so we could create hundreds of hertz responsiveness and of spiking in response to blue light pulses. By 2011, we could get red light driven spiking, in part from this Volvox Carteri derived channel rhodopsin VCHR1, in this case coupled with another mutation near the retinal binding pocket. We also were able to create these bistable patterns of spiking, so not getting one spike in response to one light flash and that's it, but you could flip cells into stable excitable states with these step function opsins that Peter mentioned as well. And that was through mutating these cysteine and aspartate residues C128 and D156 near the retinal. And then finally, we were able to, both Peter and I were able to convert the cation channel functionality into anion channel functionality. And I'll tell you a little bit more about that. And that allowed us to create inhibitory channel rhodopsins. And then we could make those bistable as well by porting on our bistable mutation on top of that. And that gave us bistable inhibition as well. And so this shows how understanding these molecules at the atomic level in great detail allows an incredible amount of power. And all of these different tools have been used to a wonderful effect in neuroscience. And amazing teams of colleagues and students and collaborators around the world have enabled this to happen. Now, crucial to it all was crystal structures, and these are the crystal structures of the channel rhodopsins that we and our collaborators were able to obtain. This is the initial cation-conducting channel rhodopsin structure from 2012 that we got in collaboration with Osamu Nareki's laboratory. And then these two, most recently, these are anion-conducting channel rhodopsins where we've been able to obtain those crystal structures as well. The initial one made it very clear that there was not a pore at the interface, but each monomer had its own pore. And this gave us an opportunity to do structure-guided engineering. What Peter and I were able to do in 2014, together with our colleagues in the labs, was to convert the internal structure of the pore from something that by itself was largely negatively charged and facilitated the flow of cations, as indicated in red here, to something that was quite the opposite, largely positively charged and able to confer all the flux of anions, in this case chloride ions. And this allowed us to create the first chloride-conducting channel rhodopsins that actually allowed you to shut off spiking with blue light instead of drive spiking. And by porting on those bistable mutations, we could create bistable inhibition. And this was done with a number of different collaborators as well. We called the advanced version of this chloride channel rhodopsin IC++. And that was one of the structures that we were able to obtain just recently and confirm both structurally what we had found functionally was that the predicted electrostatics in this protein were such that this facilitated the flux of anions instead of cations. Now, in a year after Peter and I had published the anion-conducting channel rhodopsins, as he mentioned from John Sputich's group, there was a very interesting naturally chloride-conducting channel rhodopsin. These were from the Gilardia theta, another small microorganism, GTACR1 and ACR2, and we also were able to get the structure of this naturally occurring chloride-conducting channel rhodopsin this year as well. And it was very instructive to compare these two and also to use this structural knowledge in order to create new kinds of functionality. And I'll tell you a little bit about that, again, to make the point of how important the basic structural work is. One fascinating thing about the GTACR molecules is they had enormous photocurrents, which is useful for optogenetics. It's good to have big photocurrents. One problem, though, is that they were somewhat slow, and this can be illustrated here. If one has a GTACR1, as indicated here, and delivers a single light pulse, it's not able to knock out a single spike. You have this lasting effect, and this can be important for a number of an important problem for a number of neuroscience studies. But this was something that we were interested in solving, and based on our structure, we were able to create much faster a much faster form of GTACR1. Instead of having this slow inactivation, we were able to greatly accelerate the inactivation. We called this variant flash. Now, there was another fast, naturally occurring channel rhodopsin that had been described again from John Sputich's group called ZipACR, but it had a problem well recognized in the field, including from John's work, which is this particular figure of merit for channel rhodopsins, which is this reversal potential. This is a signature of which ions are flowing through it. Both flash and IC++ and other pure chloride conducting channels under our ion balance conditions have a reversal potential indicating pure chloride flux around minus 60 millivolts. But the ZipACR was in this depolarized regime indicating that it actually had cation flux as well. And that's a big problem for optogenetic applications. For example, sometimes you can actually drive spiking instead of inhibiting spiking because of that cation flux with the ZipACR. But the flash, this faster engineered version of the GTACR1, not only had the fast deactivation, but had a reversal potential that did not allow that to happen. And in collaboration with Kang Shen's group, we put this into C. elegans, and we were able to show both by expressing flash in the muscle cells or in the motoneurons that we could suppress in reversible fashion the locomotion of the animal. So this we've seen again and again together with our teams, that getting the atomic level understanding and modeling allows not just a deep understanding of these naturally occurring proteins, but enables the creation of new tools for neuroscience. Now, another aspect of the another exemplar case of this is color shifting. And I mentioned this mutant and this mutant near the retinol binding pocket. There's an incredible diversity of these microbial opsins and their gene products in nature, and we and many others, Peter and myself and Feng and Ed, have all engaged in a great deal of discovery of new kinds of opsins, and finding new opsins has included identifying those with altered action spectrum. A particular one that was a chimera between the chlamydomonas channel rhodopsin 1 and the Volvox channel rhodopsin 1 is called C1V1. And together with our colleagues, we showed in 2012 that this allowed the first single cell control with high speed in intact living mammals. And here's an example of that. We have a patch electrode in the brain of this living mouse in the superficial layer 2-3 of cortex. So we can listen in on spiking, and here we're scanning with raster scanning this 2-photon illumination, and we don't get a spike if we're scanning just above the cell, but if we scan within the cell, we do elicit a spike, and just below we don't. And it's crucial to use 2-photon illumination for this, because that allows one to overcome, at least partially, the scattering properties of mammalian brain tissue and get that single cell resolution. Another thing that's valuable about C1V1, not only does it allow this 2-photon actuation, but it works very well in combination with optical readouts of activity. And so this is something that's become very powerful in neuroscience. A totally separate stream of technology development in neuroscience is creating these sensors. Groups in Canada and in Japan and Virginia have all greatly advanced the creation of the, for example, the GCAMP calcium sensors, which allow particularly, most efficiently, blue light excitation-driven readouts. And so having a red light control tool and a blue light readout is powerful. And together with David Tank in 2014, we showed that you could do all optical experiments using C1V1 at single cell resolution, play in and read out. And this has gone very quickly. I'm going to play a movie here. This is an example of where things are now. We and others can play in hundreds of spots of light using spatial light modulators, basically playing in holograms into the tissue. And what's shown here is this is visual cortex of an awake behaving mouse, and there are 6 successive squares here. Each of these is about 1 millimeter by 1 millimeter. But the successive squares go down from superficial to deep layers in cortex, so going down nearly 1 millimeter in a 3-dimensional space as well. And the amazing thing about integrating these fast control tools with fast readouts is you can then guide your stimulation based on how even individual cells operate in the natural case. And so in visual cortex, there are some cells that respond to vertical or horizontal gratings, and you can pick those out and then guide your stimulation to them. And here's an example of the red circles are cells that are picked out because they respond to horizontal gratings. And what we found is we can play in and control many dozens or even hundreds of those cells, and we can make the animal, a trained animal, act as if it's seeing a horizontal grating or a vertical grating by playing in just the corresponding cells and get that behavioral readout as well as the local physiology. So that's the sort of the most complicated thing you can do, very exciting and interesting, but there's also great leverage in backing off a bit, not even having to form an image. You can simply, if you know where your photons are coming from, you can just collect all the photons, and you can do that even with a simple fiber optic or other sorts of implantable optical elements. And this is a method where, for example, here we're targeting deep brain dopamine neurons in which we've co-expressed a Volvox-derived red light-driven channel rhodopsin for control and a blue light excitation-driven readout. And you can do things like give a mouse a little water reward, and you can see the dopamine neuron transient in light blue here. That's what the dopamine neurons are doing in response to something good happening to the animal. And then in that very same animal, you can try to recapitulate that with optogenetics. And Tina Kim in my lab did this experiment, and she can see she's playing in too much light here, so she can back off in light intensity until she matches exactly the naturally occurring magnitude of the signal in that same animal. And you can appreciate the value of that level of precision. The speed of it allows us to match not just amplitude, but timing of signals. Kelly Zalakuski in the lab, for example, had found using fiber photometry a very interesting blip of a signal in the nucleus accumbens that she found was crucial for guiding reward-related behavior in rats, and she was able to use that timing information to deliver a pulse at just the right time in a task and change risk-taking behavior in rats. So this is what you might call modern optogenetics. It's, we can work on any time scales. We can turn on or off with different light intensities in an easily scalable way. We can target cells or cell types, ensembles of cells in behaving animals, and we can match or modify the timing appropriately depending on what is naturally happening in the animal. And so this is where things are now, and it's kind of interesting to reflect on how far things have come. As Peter said, there were many people early on thinking about this sort of thing, how to get it working. And again, I want to just highlight how important it was to work with my co-laureates, Peter and Ed, on this. Having a fantastic team and colleagues is everything. Looking back 14 years ago, a little bit of Gairdneriana here. This is myself and Ed and Feng, and I couldn't have predicted that there'd be 3 Gairdner Laureates there standing next to each other, but that's hopefully many more to come from all of our labs, and so hopefully we'll be able to come back to this venue again. But very simple, humble experiments early on, although with their own sort of beauty, initial tests of the validity of getting membrane depolarization with channel rhodopsins. Ed's beautiful early initial recordings of spikes driven by light that really conveyed the capabilities, Feng's design of viruses and fiber optics. And all of this moved very rapidly over a number of years, but how far it's come in less than 15 years is really remarkable to think about. In the last few minutes, last 5 or 10 minutes, I'm going to tell you a little bit about where things are going into the future, at least from one perspective. And this is addressing fundamental mysteries that we've wondered about even from the very first successes that we had in controlling mammalian behavior. I'm going to play in a movie here of, this was something that happened in 2007. This was Feng Zhang's first control of mammalian behavior. And this experiment was pretty interesting, pretty simple, but it conveyed the power of the system. Even though we didn't get any scientific insight at the time, it showed the power of the technology. And so what this is a mouse that has channel rhodopsin expressed in layer 5 neurons in the brain, and we have a fiber optic implanted in right anterior motor cortex. And as you know, the right side of the brain controls movement toward and attention toward the left side of the world. And so what Feng was looking for in this experiment was, would there be any sort of detectable response, maybe a head twitch or something toward the left that could be picked up? And so what you'll see here, when the light goes on, you'll see a little bit of blue light on the head of the animal where the fiber optic is implanted. So the animal starts circling left and keeps circling left. And one more pass past the pellet and the light turns off and the animal stops immediately. A lot of people, this movie triggered definitely strong emotions in us. It also triggers some concerns, I think, in people. It sometimes is a little disturbing to see, but it also raises a lot of questions. So we know what we're doing. We know where the fiber optic is. We know which cells are light sensitive. But we'd like to know a lot more about this. We use this paradigm, we and others have used this to turn on or off cells using channel rhodopsins, or, for example, the halorhodopsin inhibition in 2010 from Alana Witten in the lab. But how is this manifested? What is going on across the brain as we're controlling the animal's behavior? And what are the opportunities for that? What if you could see the activity of all the neurons across the brain and know even a lot more about those cells? And we don't have the answers yet for how to do that. We and others are working on ways to do that. One thing you might imagine would be to take the brain of the animal and then do some sort of brain-wide analysis for an immediate early gene product like CFOS or ARC. And there are ways of doing that. We and others are working on ways of turning brains into hydrogel tissue hybrids where you've been able to link all the interesting biomolecules, RNAs and proteins, which might be immediate early gene products that would give you a marker of which cells have been active. We can link those to this sort of scaffolding as shown in these wavy green lines, a hydrogel that we build from everywhere within the cells of the tissue at once. And then we have a structure we can work with. It's transparent, and it also lends itself to ongoing chemistry and functionalization. You can even sequence, as we found, you can even sequence RNAs at cellular resolution within these hydrogel tissue chemistry hybrids, as Xiao Wang and Will Allen in my lab showed recently with a method called StarMap. And we showed actually that it does also work to identify these immediate early gene products. And so you can imagine doing that sort of experiment after a bout of behavior controlled by a specific optogenetic intervention, getting a brain-wide readout of what all the cells across the brain were doing and registering that with your known anatomy of your targeted cells. But of course, that's a slow readout of the immediate early gene products. That gives you sort of tens of minutes to hours resolution. Could we leverage the fact that we have all these cells in 3D space? Maybe we could register our live GCamp signals where we're collecting these 3D ensemble activity patterns, and could we register that with these post-life molecular and anatomical structures? That would give us hundreds of milliseconds level resolution. And the hydrogel methods do lend themselves well to this. For example, being all aqueous based, you preserve the fluorescence of these native proteins like the GCamps, and that would greatly facilitate this sort of registration, as well as ongoing sequencing. But what I want to close with is an even faster potential brain-wide readout, which is using fast electrophysiology in combination with optogenetics, and trying to get a brain-wide readout by having long shank, multiple contact electrodes and taking many different trajectories in different animals as indicated here, but having a consistent task that is comparable and then precisely timed across animals so we can integrate information on brain-wide activity patterns at high speed. So what would be the right sort of task to do? We and others have looked at fundamental survival drives. It's something that's of great interest to me and relevant to my clinical work on depression as well, where many of these survival drives, whether sleep or feeding or social interaction, are disrupted. But the task I want to tell you is the most temporally precise form of these motivated behaviors that we've been able to get working, which is very important in this case, and this is thirst, a thirst-related behavior. About a year ago, Will Allen in the lab, and this is in collaboration with Li Chun Lo, we were able to show some key neurons deep in the brain, in a deep brain structure called the MNPO, that are crucial for thirst-related behaviors. And since it's published, I won't go into the genetic targeting strategy. Suffice it to say that we were able to express this engineered chloride-conducting channel rhodopsin, IC++, only in cells that had been strongly active when the animal was thirsty. And so we had a method of doing that. So we gave the animal a period of time when it didn't have access to water for a little while, was moderately thirsty, and we were able to drive expression of a channel rhodopsin only in those cells that are strongly active during that time. Then we could come back later and ask what happens if we turn those cells up or down. And we found that inhibiting those cells, we could, even an animal that's very thirsty, that's licking hundreds of times in the behavioral epoch, we could powerfully suppress its licking for water using IC++ in those targeted neurons. And that was pretty amazing to see, a very fundamental, powerful survival drive being so precisely suppressed in that way, and control animals that had the same sort of expression, but only driven by cells that were active when the animal was in its home cage, not thirsty, did not have that effect. That was suppressing activity. What if we provide activity in those neurons? We could drive licking behavior, even fully sated mice. Here are 5 separate mice, onset of the driving the excitatory channel rhodopsin in these MNPO cells, and driving intense licking for water, even though these animals are fully sated, that terminates when the light turns off. And we also were able to show that this was an aversive state. The animals avoided a chamber in which they were getting this stimulation and spent a lot of time as far away from that chamber as they could. And we all know thirst is an aversive state as well. So many features of the state were recapitulated by driving this deep local population of cells. But to come back to this question of getting a brain-wide readout, we wanted to design a temporally precise version of this task so we could compare, first make use of the high speed of the NeuroPixels probes and also compare across animals with everything time-locked. And this is a task that Will Allen in the lab developed. It's a go, no-go task. You have mice that are trained to know that one odor predicts that there will be a water reward coming, and they lick at the little spout for water. And they know that another odor predicts there will not be a water reward coming. So it's a go, no-go task, and the animals learn this very quickly. And here's an example of that sort of behavior. Here's a thirsty mouse receiving the go cue interleaved with no-go cue trials, and you can see licking intensely after the go cue, but not the no-go cue, but stopping upon reaching satiety. Okay, so that's the behavior. Now, question is, very simple question, what's happening across the brain while this is happening? What is the brain-wide state of being thirsty and trying to get water? And by the way, these 3 vertical lines here are recapitulated here, onset and offset of the odor cue, and then onset of the reward. And so this is go cue, no go cue, and each of these are color-coded brain regions across the whole brain. And what we have here horizontally are many thousands of rasters indicating individual neurons, what their spiking pattern is during this task. And red means higher activity, blue means lower activity. And the first amazing thing was that virtually the entire brain is engaged by this task, and most of the neurons in the brain. So more than half of all the neurons in the brain are clearly engaged with this task of acquiring water when thirsty, which was a remarkable result. And you can see the clear distinction between receiving the go cue and the no-go cue. These are equally thirsty animals. Okay, now, big question. What happens when we're providing the thirst state with optogenetics? What happens? Okay. And so here we provided optogenetically the input to these MNPO neurons that I showed you before, and you won't be surprised to see this is what happens. The mouse that licks to satiety, and then you drive the input to these cells, and you get robust licking behavior again. So what's happening across the brain while we're doing this? And this was the amazing result, is that we're able to recapitulate virtually the entire brain-wide state with high temporal resolution and in ways that match at the detailed level right here. This is a sated animal receiving the optogenetic stimulation with a nearly identical brain state compared to the truly thirsty animal. So here we have now a brain-wide understanding at cellular resolution of a state that, and we have control over it, and we know which cells are able to recruit it. And this is a remarkable level, I think, of insight and understanding. And just to think how far things have come since 1866, but also how important basic science is. I'm sure Andre Fermenson would not have predicted where things have come. It took us a little while to get there, but things have gone very quickly in recent years. A final note, people often ask about clinical implications. Peter mentioned this briefly. It's not something I'm working on in the lab, but people think about it and talk about it. And I think a key principle is the concept of the optogenetics guided clinical intervention. So this is, you don't have to think about utility of optogenetics, which is primarily a basic science tool, the basic science discovery tool. But any insights that come from that will make any kind of treatment more powerful in human beings, and this is a good example. Anto Banchi, who studies addiction, had found in 2013 that he could turn down optogenetically the cocaine-seeking behavior of cocaine-addicted rats, and it was such a powerful effect that he designed and launched a clinical trial using transcranial magnetic stimulation, armed and guided by the causal knowledge from optogenetics, and they were able to see a powerful reduction in this controlled study of cocaine-seeking behavior in human beings with cocaine addiction. They're following that up with a larger, longer-term study, but it's an example of the optogenetics guided clinical trial, which I think is an interesting concept. And again, coming back to the basic science question, understanding the fundamentals of the proteins themselves, even down to the atomic level, has been crucial for us in getting this far. So I'll wrap up there, and again, a deep congratulations to my fellow laureates, Peter and Ed, who are really wonderful and creative and brilliant folks, and here in Gairdner Red, the fellow Gairdner laureates who contributed to this, and thank you for your time. Thank you.